Hi, Tam here. Welcome to our third lesson for this week, which is protein trafficking. So our learning objectives for today are to be able to recall protein structure and function, understand and describe how proteins link organelles during protein trafficking, and then apply your understanding to exam questions. So we're going to start off with a really quick quiz. So just jot down what you think the answers are. So I'm going to give you some clues. As soon as you think you've guessed it, jot down your answer and then we'll see if you're right. Okay, so what am I? I'm a protein. I am globular. I have a quaternary structure. I'm made from 4 polypeptide chains. I have 4 iron groups. I'm found in red blood cells. And my last one, 4 oxygen molecules combined to 1 molecule of me. So you might have guessed I am hemoglobin. And there's the structure in case you forgot. Okay, what am I? I'm a protein. I'm globular. I have disulfide bonds. I can have a tertiary or quaternary structure. I am released by plasma cells. You'll learn more about those next year. Plasma cells are a type of white blood cell. And if a bacteria infects your body, then your immune system will release these to attach to the bacteria's antigens and aid in the destruction of the bacteria. So you might have guessed it's an antibody. Okay, what am I? I'm a protein. I am a structural protein. I'm fibrous. I'm made from many alpha helices. I'm a key structural material making up the outermost membrane of human skin. The only other biological molecule that is as strong as me is chitin. And lastly, I'm a key structural component of hair and nails. Hey, so if you got keratin, well done. There it is. Please pause the video and complete activity 1, recall. So just to review this then, the nucleus, as you guys know, is where the DNA is stored, and the DNA codes for proteins, where it has lengths of genetic material called genes that code for polypeptides. We'll come back to that more later when we look at protein synthesis. The rough endoplasmic reticulum, so this is where proteins are folded into their 3D shape, and they're also transported through the cell. The Golgi body, this is where modification of proteins happens. And the cell membrane, so if proteins are released from the cell, then they can be released via the cell membrane. Please pause the video, complete activity 2. Okay, so just as a quick reminder, if you didn't already watch this video, please go back and watch that before you carry on with this video. It's a really good overview of what happens in protein trafficking. Now, protein trafficking is basically how proteins are synthesized and then modified and moved through a cell. So just as a reminder, in the nucleus, which is up here, you have DNA. Now, DNA has lengths on it called genes, and the genes can code for polypeptide chains, which then can form proteins. Now, if you remember from GCSE, copies of those genes called messenger RNA are synthesized, and those messenger RNA molecules can travel out of the nucleus via a nuclear pore, and they'll only ever go out, they never go back in. So we have to be careful when we're describing that in exam questions. And the messenger RNA will move to a ribosome located on the rough ER. And on that ribosome, the messenger RNA is going to code for a polypeptide chain. Now, once the polypeptide chain has been synthesized, it's going to move through these flattened sacs in the rough ER, and it's going to be folded into its 3-dimensional shape. Okay, and once it's moved through those flattened sacs, it will bud off in part of that membrane as a vesicle, and that now 3-dimensional folded polypeptide chain can move to the Golgi body, and here is the Golgi body. And the Golgi body is where modification is going to happen. So this little vesicle is going to fuse and that's quite a good word to use here, with the Golgi body, and the polypeptide might be modified. So what can happen there is something like a carbohydrate chain might be added to form like a glycoprotein, and then that protein might have a particular function. Or if, for example, it was hemoglobin, then it would be a prosthetic iron group would be added, and you'd have 4 polypeptide chains combined together. So modification can be adding more polypeptide chains, or maybe adding a prosthetic group that is something not made of amino acids. So once the protein has been modified in the Golgi body, it can then move out of the Golgi body within a vesicle, okay? And these vesicles will transport the now modified protein to the cell surface membrane potentially. And if it's being excreted from the cell, then it can leave the cell, or maybe sometimes it will be moved to elsewhere in the cell. So here's a little bit more detail about that. So you might want to pause the video and just check your answers for activity 2 and add any details that you need. Okay, please pause the video and complete activities 3 and 4. Okay, so just to review one of these questions with you, we're looking at this question here. And one thing just to notice is that this diagram might look a little bit different from ones that you've looked at before. This is a ribbon diagram, and the thing that looks a bit like a ribbon is the polypeptide chain. So there's lots of different ways of modeling and looking at protein structure, and ribbon diagrams are one of them. So a couple of things to notice here. First of all is the groups. Okay, so you can see here we've got the amine group and we've also got the carboxyl group. Okay, now what that sort of implies to us when we're looking at this diagram is that this is one polypeptide chain, because we've only got the start of one end here, the amine group, and the other end of that chain would have the carboxyl group. Now, if this was a quaternary protein, we'd have more than one polypeptide chain. So we'd see another chain with another amine group and another carboxyl group. So we know here that this is a tertiary structure, and we know that the tertiary structure is when the secondary structures are folded, and we can tell that it's not a quaternary structure because we've only got one polypeptide chain. As your plenary, have a go at this Kahoot. And just finally, a few of our learning objectives to quickly review and see how confident you feel about this lesson. Thank you.